Hello, everybody. Apparently there was a error of communication somewhere, or everybody was really busy. But so that you guys don't get behind, I'm going to go ahead and lecture on the two chapters that are on the first week's module and make this available to anybody who wants to watch it, so at least you can hear what I have to say. So I'm going to go ahead and get started. We have chapters 2 and 24. So what you want to do before you continue with this video is get those chapters printed out and have them ready so that you can fill it in as we go. Since this is a recording, you'll be able to pause and restart this video whenever you want, so you can move at whatever speed you want. I'm going to go ahead and show chapter 2. Okay, this is chapter 2, chemical level of organization, reactions, and enzymes. Okay, some of the main concepts that we talk about at the beginning is energy, enzymes, things like that. So energy in the form of ATP does all the work in our body. Chemical work is the ability to synthesize new molecules or break down large molecules. Notice that the words in green correspond to the blanks in your lecture notes. That is true for most, or at least more than half of the chapters. And then later on, I didn't continue doing that. Okay, transport work is the ability to move molecules across membranes around within cells. Notice that you need ATP to do all of this work. Mechanical work is flexing your muscles or moving cilia or flagella. We have flagella in our sperm cells and cilia in our respiratory tract. Okay, some review of chemical reactions. In a chemical reaction, if there's A plus B, these are the reactants, and C plus D are the products. So A plus B is the reactants, and C plus D are the products. The arrow symbol indicates a chemical reaction has occurred. So if I ask the question, what symbols do you usually find in a chemical reaction? These A, B, C, and D are variable, but you usually find plus symbols and arrows. So that's the kind of symbols that you find in most chemical reactions. What are some different types of chemical reactions? Well, A plus B turning into C is a combination reaction. C turning into A plus B is a decomposition reaction because it's decomposing from something large into two small things. L plus MX turning to M plus LX. You can see it's single displacement because L took the place of M in that molecule. And then here you can see they both switch partners. So L and M switch partners, and it's called double displacement. Energy in chemical reactions. There are two basic types of chemical reactions based on the energy released. The first is called endergonic, and that stores energy in chemical bonds. A good example of that is photosynthesis. Same kind of thing would be happening if you were building muscle tissue or even storing sugar in the form of glycogen in your liver. Exergonic is the opposite. This is energy releasing. When ATP is being used for cellular work, energy is released, and so these are opposite from each other. Okay, this is a figure directly from your OER text. This shows how enzymes work. We have substrate 1 and substrate 2 coming together in the enzyme in the place called the active site. And then notice that these two substrates form a product, which is now a single structure, and then it's released, and this process repeats. Enzymes are what are called protein catalysts. They're made out of protein, and they catalyze chemical reactions, which means to speed them up. They're usually named for what they do. So you can look at the name of an enzyme and tell what it does. So the suffix ASE is usually added onto enzymes, but not always. There's a few that we'll talk about that don't have that ASE ending. By the way, OSE, like glucose and fructose, is the suffix for sugar. A catalyst is any molecule that causes a chemical reaction to occur, but does not change itself in the process. So notice the enzyme in step D is the same as enzyme in step A. The active site of the enzyme is the same shape as part of the substrates. They change. Notice that the two substrates have equivalent active site shape opposites, so they can fit in. If they don't fit exactly, they won't fit together, they won't be converted, and the enzyme has no effect on those substrates. Enzymes speed biochemical reactions. So now talking about inside the body. So what do? They lower what is called activation. So what is activation energy? Activation energy is the amount of energy that you have to put into a chemical reaction to make it go. For example, when you burn paper, the fire is the energy that you add to cause the chemical reaction to light the paper on fire. So enzymes, they cause reactions to occur more quickly. They can cause over 10,000 chemical reactions to occur in just 1 minute. Your notes may say 1 second, so please change it to 1 minute. Enzymes may need to be activated. Enzymes aren't always active. Sometimes they need to be activated. Enzymes involved in blood clotting and digestion need to be activated. So if it's an inactive enzyme, usually it has the suffix ogen. For example, pepsinogen becomes pepsin when it's active, and then it digests protein. For checking your understanding, why is it important that enzymes not be active at all times? Well, you don't want them to be active in the cells that produce them, and you only want them active when it's appropriate. For example, you don't want the proteins in your blood to clot unless you have an injury where you're bleeding out. Some enzymes require cofactors or coenzymes. Some of these are vitamins. Some vitamins act as coenzymes. Okay, modulators. Enzymes function best at ideal conditions. That is, what temperature and pH they prefer. Changing the ideal condition slows or stops enzyme function. So if they don't have ideal conditions, they won't move as quickly, they won't cause as many chemical reactions to occur. What are some modulators? Temperature. So temperature, we're going to see, can speed them up, and it can also slow them down, and it can destroy them. pH, for example, the acidity or basicity, each enzyme likes its own particular level of acid or base. We're going to see that here in a moment. And inhibitors. Now, inhibitors are things that can stop enzymes from working, that can either be permanent or temporary. Okay, the ideal temperature for any enzyme is specific to that enzyme. For example, most of the enzymes in our body prefer 98.6. The exception to that is sperm cells that prefer slightly cooler, and that's why they form outside the man's body instead of inside. So that's part of the reason why they are not inside of our abdomen, but on the outside. If the enzyme is too cold, it moves too slowly and does not cause as many chemical reactions to occur. This is how hypothermia occurs. If you're hypothermic, the chemical reactions are not occurring quickly enough, you're not generating enough energy, and you die from that. When the enzymes are heated beyond their ideal temperature, the active site starts to change shape. If the active site is no longer proper shape, can't interact with the substrates, can't produce energy, and this is what happens with fever or heat stroke. The enzymes start to change shape, and they can no longer function. When the enzyme's active site is permanently changed, the enzyme has been denatured. This is the term I'm going to want you to know. It means that the shape of it has been bent, and it can't be bent back. pH. The pH of a liquid can determine how well an enzyme will function. That's the acidity or basicity of the liquid. All enzymes have an ideal pH at which they best function. So imagine the enzyme that is in your stomach, pepsin, likes acid, although the one called trypsin in your small intestine likes neutral. So each has their own pH that they like best. Changing the enzyme's pH causes changes in the shape of the enzyme active site. So in the same way that heat can cause the active site to change shape, so can non-ideal pHs. Now, cold does not denature enzymes, it just slows them down. Severe pH changes cause the active site to become denatured, and the enzyme cannot work. It's permanently broken, you could say, and it has to be destroyed and reformed inside the body in order to be functional again. Okay, so this shows you a figure. This is not from your book. This is something I found from another text, but it shows you low to high enzyme activity. That means how fast it works, how well it works. You can see for each of these enzymes, there's a pH that it likes best. So for example, pepsin is in your stomach. That's acid because we have hydrochloric acid in our stomach. Amylase found in the mouth. Your mouth is neutral. Trypsin is found in your small intestine, slightly alkaline. The alkaline is important because that is used to neutralize the acid coming from the stomach into the small intestine. Enzyme inhibitors. Now, inhibitors means that it slows down or stops the enzyme. Competitive inhibition occurs when a molecule that's close enough to the shape of the true substrate will fit into the active site. So it blocks the active site from accepting the substrate. The inhibitor molecule prevents the true substrate molecule from getting into the active site, thereby stopping the enzyme from doing its job. So for example, cyanide ions completely block the active site of the enzymes responsible for cellular respiration. And this is why cyanide kills you, because without enzymes, you cannot generate the energy you need to stay alive. And if cyanide is blocking those, then you can't release the energy you need to stay alive and you die. How does cyanide kill a person? Well, it blocks the enzymes that give you the energy, ATP, to make you alive, and you die because your enzymes aren't working. Now, a different kind of inhibition is called non-competitive or allosteric. Just to let you know, because there's two different terms here, there may be two correct answers on the test, and I'll be talking about that tomorrow in lab. I'll be going over the policies, ways to be successful in the class. But keep your eye open for non-competitive and allosteric, both being correct answers on the first test. They attach themselves to some portion of the enzyme molecule close to the active site, but not in the active site. And what that does is, because they're attached somewhere else, the active site changes shape and can no longer accept the substrate, and therefore it doesn't work. Heavy metals like lead, mercury, and chromium. Now, these can be removed with chemicals called chelating agents, and once you remove them, then the enzyme can continue to function. This is part of the reason why lead poisoning harms you, is because it destroys enzymes. Okay, so this shows you how this works. This is not from your text, but it's just an image that shows the competitor fits into this little spot here. So now the substrate can't fit in. Now, in a different kind called allosteric or non-competitive, you see the allosteric inhibitor coming in. What that does is it causes the active site to change, and still now the substrates don't fit in and the enzyme has been inactivated. Now, your body does this on purpose sometimes. It's not always a poison. Your body can do this so that the enzyme is only active when you want it to be. And the analogy I'm going to use is an ice maker. We don't want ice makers making ice 24 hours a day because it would push your freezer open. You only want to have it make ice when you need more. And when you have a full bin, you want it to stop. So sometimes enzymes are inactivated because you have plenty of the product. You don't need it. Okay, cell metabolism. What is metabolism? It's not just breaking down food. It's the sum of all chemical processes through which cells obtain and store energy. So all the chemical processes in our body is called metabolism. Normally, we just say, how fast do you process food? That's not entirely true. There's two types of metabolic reactions. The first is called catabolic, energy releasing. Now remember, energy releasing, we saw that earlier, right, with exergonic, right? So catabolic and exergonic go together. And what that does, it breaks large molecules into smaller ones most often. Notice anabolic is energy requiring. This would be endergonic. So anabolic and endergonic go together. This is energy requiring, and it usually makes small molecules into larger ones. The way to remember that is anabolic steroids help build muscle. They take smaller amino acids, make it into protein, which becomes your muscle. So anabolic steroids are used to build muscle. So anabolic or anabolism means to build, catabolism means to break down. So we do this primarily in our digestive tracts. We do this in various parts of the body when we're trying to build molecules. In our liver when we're building glycogen, and in our muscles when we're exercising. They also can convert one molecule into another. Okay, this is feedback inhibition. Now notice A goes to B by enzyme 1, and then B goes to C by enzyme 2, etc. Now notice when we get to Z at the very end of the process, Z goes back, it feeds back to the beginning and shuts off this enzyme because the body has enough Z and you don't want too much. In the body, homeostasis is the key term for all of physiology. And homeostasis means having just the right amount of everything, right temperature, the right amount of food, the right amount of everything. So if you have plenty of Z, you don't want more, you want just the right amount. So what Z does is it goes back and shuts off enzyme 1, prevents it from working. And then when you don't have enough Z, falls out of here, and then you make more of it. And that way you always have the right amount. This is a good example of, or the best analogy is the ice maker. When you take some ice out, the little arm drops down, that stimulates the ice maker to make more ice. And then when it fills up, it senses that the ice is full, it shuts off. So interestingly, it's the ice that actually shuts off your ice maker in the same way this product shuts off the sequence early in the process. So it's called feedback inhibition because Z is inhibiting enzyme 1 and it's feeding from the end back to the beginning. So for example, you give me a paper, that's you giving it to me, and then I give you feedback by sending it back to you to the beginning, which is you creating it, and then you get feedback on what you did. So feedback inhibition is the presence of the end product, in this case Z, of a series of enzymatic steps cause the process to stop temporarily. The end product acts as an inhibitor to the enzyme early in the sequence, so it goes back and turns it off temporarily. Okay, so that was that chapter. And so now we're going to go ahead and jump to chapter 24. So please get that out. And here we go. Chapter 24.1 and 24.2, making ATP. And you can see that that corresponds to a later chapter. The way that my original book was set up was different. And in your current OER text, this particular process is described way later in chapter 24. I know we're going to be jumping around a bit, but if you want to look it up in the text, it's chapter 20. Okay, making usable energy ATP. We talked about this already. The ATP is the molecule the body uses to make energy. Although energy is stored in the bonds inside food molecules, this energy is not available to cells for work. So the way to think about this is food molecules are kind of like a paycheck, and the ATP is like cash. So if you took a paycheck into a 7-Eleven and wanted to buy something with it, the clerk would say you need to cash it first. So this process we're going to be talking about today is how your body takes this paycheck, which is food, and converts it into ATP, which is cash. And that cash is what is spent in the body to do energy requiring jobs. ATP is a form of energy the cells can use to do work. So this is really the only form of energy that the cells use to do work. There's a couple of exceptions we'll talk about later in this chapter. ATP stands for adenosine triphosphate. So tri means 3. Phosphate is a molecule made up of phosphorus and 3 or 4 oxygens. When the ATP does work in the cell, one of the phosphates comes off, turning the ATP, which is 3, into ADP, which is 2. Di meaning 2. So one of them falls off. It doesn't disappear. It's around, but it's just no longer attached. And another good analogy of that is that the ATP is like a charged up battery and ADP is a dead battery. So if you want to make a dead battery useful again, you have to add energy to it. In this case, adding another phosphate turns this dead battery, ADP, into the charged up battery, ATP. When food is broken down, the phosphate is reattached, energy is stored, and ATP is ready to do cellular work. So now you've redone it. So this is a figure from your text, and it shows ATP being the byproduct of the breakdown of glucose. So when glucose is broken down, you make 2 molecules of pyruvate, which is a precursor to the process we're going to be talking about later. When you do that, you get a little ATP out of it. And later on, we're going to see that the pyruvate goes into this next process in the mitochondria. Carbon dioxide is removed and it becomes what is called acetyl-CoA. So pyruvate becomes acetyl-CoA when you add a coenzyme A to it. Then eventually that's going to go into the Krebs cycle. We're going to be releasing carbon dioxide atoms. That's why we breathe out. That's what comes out of our breath when we breathe out, making more ATP. And then eventually our last process is called oxidative phosphorylation or electron transport. We have a better figure showing that this is just kind of an overview to let you know where we're going to be going with the rest of this lecture. Okay, so this shows you the overall equation for cellular respiration. So what a common fuel for cellular respiration is glucose. We can burn all different kinds of food molecules, but glucose is a common one. So this is the formula for it, C6H12O6 plus oxygen, and we get that by breathing in, and it's converted into carbon dioxide, which comes out of our mouths when we breathe out. Water, we actually make a little bit of water. I'm making water right now just by being alive, and the main product is ATP. This is the overall equation for what happens to glucose during cellular respiration. Please note, if you don't see these numbers out front, the 6, 6, and 6, it's still a proper chemical equation. It's just not balanced. So if you don't see these numbers, these coefficients, these big 6s, still correct, and I'll show you. Okay, the formula for glucose again is C6H12O6. When sugar is broken down, the energy in the molecules bonds are converted into ATP energy. The byproducts or wastes, CO2 is definitely waste, but water is something that we use in our body, so it's good that we make a little bit. We make a little tiny bit, not enough to keep us alive. This is an example of an unbalanced chemical equation. Notice that there's no 6s out front. This shows you just the sugar and the oxygen, carbon dioxide, which is our waste that we breathe out, and water that we make. So if you had to put this in the words, you would say the sugar from your food that you eat, plus the oxygen that you breathe in, is converted into arrow, carbon dioxide that you breathe out, and water, which you keep with ATP energy as the main product. The point of this is you're making ATP. Notice it's not a main ingredient in this. It's more like it happens as a result of the breakdown of this. It's not a direct product of this specific reaction. Cellular respiration and breathing are closely related. You have to be able to breathe to do respiration, and you have to have energy that you made during respiration in order to contract the muscles that allow you to breathe. Cellular respiration requires the cell to exchange gases with its surroundings. So for example, even a bacteria has to do cellular respiration. Breathing exchanges these gases between the blood and the outside air. So we breathe in, the oxygen goes into our blood, the blood moves to our body tissues, especially our muscles, it offloads it to our tissues, and then our tissues use the oxygen to make energy. So here's a good image. This is not from your book, from a different book, but you can see oxygen coming in, going from your lungs down to your muscle tissues. Then cellular respiration occurs. What comes out of it is carbon dioxide. It makes its way back to the lungs, you breathe it out. So you breathe in, you're taking in oxygen to your cells. Your cells use it, convert it into CO2, which you then breathe out. Okay, so respiration is two ways. So we're going to be talking about two men in the upper slightly left and two men in the upper right. You've noticed that these two men here are resting. No, they're not sleeping. This guy can't sleep standing up. They're resting. And these two guys are running. What I want you to notice about these two guys is they're running as fast as they can. You can see their expression. They're not doing a half mile. They're not doing a marathon. They're sprinting. You can tell from their expressions and their basic physique that they're sprinters. What this means is they're going as fast as they can go in the moment. And the key is they're going to have to stop pretty soon. Sprinters can't sprint forever. After a few moments after this picture is taken, somewhat up to 10 seconds, they have to stop. These two guys can do this all day, and I know some people who do. All right, anaerobic respiration occurs in the cytoplasm. That means the area between the organelles and your cells, and does not require oxygen. Another term for that is glycolysis, which is sugar splitting. These two men in the upper right are doing what is called anaerobic respiration. Now, that doesn't mean they're not breathing. What it means is the amount of energy that they need is not being provided by their breathing. So they're having to go into what is called anaerobic respiration. And that is a less efficient strategy. And you know it's not going to work for very long because they're going to have to stop and switch back to this way at the end of the race. And means without, and aerobic means oxygen. So in this case, no oxygen is required to do this. Now they're still breathing and there is some oxygen, but it's not enough to give them the energy they need to sprint. Sprinting is an anaerobic activity. That's what these two guys are doing. What is the upside of this? Well, you don't have to have oxygen. If they are trying to generate energy and there's not enough oxygen in their cells, they can still keep going for a little while, enough to win the race or escape an animal that's chasing potentially. All right, what's the downside? Well, you only earn 2 ATP molecules per sugar, which is a really terrible yield, but it's better than none. And the other problem is you build up lactic acid. So if you've ever been sprinting or doing something that's really, really intense, you start to feel the burn. And you have an example of that. I'm starting to feel the burn. What that means is as soon as you feel the burn, you switch from aerobic to anaerobic. And the burn comes from the lactic acid accumulating in your tissues, especially your muscles. Aerobic respiration occurs in the mitochondria. So instead of between the organelles, it's in the mitochondria. We're going to look at that later, and requires oxygen. So these guys, in order to do this, and you watching this video, you have to have plenty of oxygen to do this. So when you're at rest, you're burning sugar aerobically. And I know you're at rest because most people aren't going to be sprinting when taking notes in my class. What is the upside? Well, look at how much better a yield is it per sugar. You get 32 ATP molecules per sugar, and so it's a much better strategy. What's the problem? You have to have plenty of oxygen in your cells. So if you're not at rest or doing light exercise, you can't do this. You have to switch over to anaerobic. Now, you can walk for hours, but you can't sprint for hours. So these guys have switched from aerobic to anaerobic, and we know pretty soon after this picture was taken, they're going to have to stop and rest and switch back to this. Okay, fermentation is another term for anaerobic respiration. You might want to note that if that's not in your notes, write that down. Anaerobic respiration in human muscle cells. Okay, human muscle cells have enough ATP to support activities such as a quick sprint for about 5 seconds, but we don't stop after 5 seconds. We can keep going, most people. So the secondary supply of energy is called creatine phosphate, and that'll give us another 10 seconds. When those two are gone, then you have to switch to anaerobic respiration. To keep running, your muscles must generate ATP by the anaerobic process of fermentation, the other term for anaerobic respiration. Now, you know you've switched from aerobic to anaerobic because you start to feel the burn of the accumulation of the lactic acid. It's actually burning your cells. The product of lactic acid is fermentation in humans and other mammals. This is what we do. Not all living things do it. Fermentation in microorganisms. Some microorganisms do it differently than us. And this is good if you want to drink beer or wine or any kind of alcohol. So for example, yeast. You guys have heard of yeast? You mix yeast with hops and barley, wheat. You're going to make something different. You're going to make CO2, which is the bubbles in beer, and alcohol, which makes it alcoholic. So instead of making lactic acid, there's not enough oxygen, they make carbon dioxide, which is the bubbles, and alcohol. And when they've developed enough carbon dioxide and alcohol, it actually kills the yeast, stopping the process. And that's why beer usually has about 4% alcohol, because that's enough to kill the yeast cells. And now you've made the beer right in its bottle. It's drinkable. Okay, this is a figure from your book that shows how it works. This is very complex. I'm not going to ask you to know all of it, but if you do look in the book, you'll see that we're talking about On the right side, anaerobic, because notice that we have lactate here. Please note and write this down. Lactate, the ATE ending, is the same thing as saying lactic acid. So in this case, pyruvate, what would that be? Pyruvic acid. So pyruvate, ATE ending, is the same as saying ic acid. Pyruvate, pyruvic acid. Please keep that in mind. On the left side shows what happens if we have plenty of oxygen. So this is aerobic on the left, anaerobic on the right. If you have plenty of oxygen, you do this. If you are sprinting, don't have enough oxygen, you do this. Okay, so this is in your book. So the first step is called glycolysis. This takes place in the cytoplasm. As I said, that's the liquid part of the cell between the organelles. The 6-carbon glucose is broken down into 3-carbon molecules called pyruvate or pyruvic acid. During this breakdown, 2 ATP molecules are produced. So it's not a very good yield, but you don't have to have oxygen. But the key here, one of the keys I want you guys to know, is that NADH are also produced to carry hydrogen. This is what is called a hydrogen carrier. So I want you guys to focus on NAD being the wheelbarrow in this analogy, and the hydrogen is what is being carried. So NAD is the carrier, hydrogen is the material being carried. Oxygen is not needed for this step, so it's called anaerobic, and this is the sprinting. So this shows that we're starting off with sugar. We're going to make a ATP. Now, what I want you to see here is notice the ATP is becoming ADP here. So what we're actually doing is we're actually investing energy at the beginning. In the same way it takes money to make money, you have to buy stuff if you're going to sell it to make a profit. In this case, the body invests ATP at this part and then gets yield out at the end. So if you count the yield at the end, 1, 2, 3, 4, but subtract the 2 that you had to use up at the beginning, you get a total of 2. So if you spend 2 and earn 4, the total is 2. Okay, and they're showing you some simplification of how this works, but the key here is you've got pyruvate molecules that will either enter aerobic respiration, be broken down to carbon dioxide, or turn into lactic acid if you don't have the oxygen. Okay, there's 2 terms for this next process that we're going to be talking about. It's called the Krebs cycle, traditionally, and also sometimes called the citric acid cycle. Why is it called the citric acid cycle? Because the very first molecule that's formed when the acetyl-CoA comes in from the Krebs cycle prep stage, the very first molecule formed is called citric acid. It's the same molecule we find in lemons and oranges, citrus fruits. That's why it's called citrus, because it's citric acid. In the Krebs cycle, the pyruvic acid from glycolysis is prepped or prepared to go into this Krebs cycle, and it becomes a molecule called acetyl-CoA. Acetic acid plus coenzyme A becomes acetyl-CoA. The pyruvate or pyruvic acid, as I told you, enters the mitochondria if there's plenty of oxygen present. What's going to happen along this process, this cycle, is that carbon atoms are going to be removed. And by the way, if it asks the question, what former molecule are all the carbon atoms removed? It's carbon dioxide. That's how all the carbons are removed from the initial molecule, from that acetyl-CoA or pyruvate. Pyruvate then becomes 2-carbon acetyl-CoA, and NADH is formed to carry hydrogen. This is part of the prep. Okay, so we're going to make acetyl-CoA, and eventually it's going to enter the Krebs cycle proper. The acetyl-CoA attaches to a 4-carbon molecule, making a new 6-carbon molecule. That 6-carbon molecule is right there, citric acid. So this is the prep thing right before the citric acid or Krebs cycle. And this 6-carbon molecule is citric acid, which is the first main molecule of this process. Another carbon, as carbon dioxide is removed, notice that the way that we're getting rid of the carbons that came in is in the form of carbon dioxide. Now we make a 5-carbon molecule. You don't need to worry about that. But look, here's another one of those hydrogen carriers. There's one here. There's one here. We saw one in glycolysis. So the key is these are going to end up going to our very final process called electron transport and dumping these hydrogens so that you can use those hydrogens and the energy carried within this molecule to make ATP energy. It happens again. Now we're going to release one more CO2. That should be the last carbon that came in, make another hydrogen carrier. But here's something I want you to note. ATP is formed, but only one molecule. The main purpose of the Krebs or citric acid cycle is not to make ATP directly. It's to make these hydrogen carriers. See them? 1, 2, 3. Then eventually we're going to see another one. There's another NAD H. But we also have what I call a double wheelbarrow, because instead of carrying one, it's carrying two. So it's FADH2 instead of NADH2, but the idea is the same. It's carrying hydrogens. So this is what I want you guys to know. The main point of the Krebs or citric acid cycle is to make these hydrogen carriers, not to make ATP directly. And that's one of your review questions. Okay, this is showing how it works. This is the details of it. So remember at the top here, we have citrate. If you guys remember, citrate and citric acid, it's the same thing. And what it's showing you here is all these molecules. You don't need to know that, but the key I want you to know is there's carbon dioxide coming off. There's carbon dioxide coming off. Where's the other carbon dioxides coming off? I don't think they're showing all of them. But the key here is I want you guys to see ATP being formed here, hydrogen carrier being formed there, hydrogen carrier being formed there, hydrogen carrier being formed there, hydrogen carrier being formed there. So the key to this, the key is what's the key product is these here, these NADH, several of them, and the FADH2. So these are going to end up going to electron transport, dropping off their payloads, and that'll be the end of it. Okay, so now we're to the last process. This is called electron transport. Most of the ATP a cell makes is an ETS, so very little has been made so far. What we're trying to do is get all these molecules that we've been making to the end so they can make lots of ATP. All the NADH and FADH2 reach the ETS and dump the hydrogens that they are carrying. In addition to dumping hydrogens, they're also dumping electrons that have a lot of energy. And we'll be talking about what happens to these electrons and how that powers the system. When this happens, the electrons in the bonds, that is between the NAD and the hydrogen, there's electrons in there connecting. They move through the protein chain. The electron transport chain. As the electrons flow, it's like electricity. You can do stuff with electricity. In this case, we're going to be pumping hydrogens. The flowing electricity is going to act like a pump, moving hydrogens from where there's fewer of them to where there's more. This is the opposite of diffusion, which says when there's a lot of concentration, you open the gates, it's going to tend to spread out. In this case, we're going to be moving hydrogens from where there's less to where there's more, and that takes energy. The moving electrons cause hydrogens to be pumped through the membrane against the gradient. That means the way that they don't want to go. You're using the electrons as a way to push them the way that they don't want to move. Now, you have a whole bunch of hydrogens on one side of the membrane, very few on the other. You open the door, you make 32 ATPs as they move through this molecule that I'll show you in a second. So it's the flowing hydrogens that have been pumped by the electrons, the electricity, that are actually making ATP. The moving electrons have to have a place to go. And those hydrogens that came with them, they need a place to go as well. So what happens is they hook up with oxygen. And when you take oxygen and hydrogen and electrons, what do you get? Water. So this is why we make water in our bodies, because the oxygen that we're breathing and those hydrogen that came in from the Krebs cycle have to form water. And I'll show you why they have to form water when we get to the figure. The key here is now these ATPs can do cellular work, and this is what keeps us alive, all this process. So the key is these dump the hydrogens and electrons, the electrons pump the hydrogens from where there's less to where there's more, and then when the hydrogens flow back through the way that they want to go, it causes thing to move, and that makes ATP. Now we got what we need to be alive. Okay, so here's a figure from your book showing how it works. So let's take a look. Here's and this is likely to be an essay on your first test. And I'll be talking about how to deal with the essays tomorrow in our first lab. Okay, there's the NADH from the from Krebs cycle or citric acid cycle. Notice what it's dropping off are these hydrogens. So now it becomes NAD plus hydrogen, so it drops it off. The other thing that's dropping off are these sunburst shapes, which are electrons. So it's dropping off hydrogens and electrons. And then if you watch the flow, this yellow line is going through these cytochromes. And as it flows, it pumps hydrogen from here to here. Notice how many there are up here. There's already a bunch, but it pumps it from where there's less to where there's more. That's what the electrons are doing. Now, where do the electrons go? For the same reason why you have to connect both sides of the battery, these electrons have to have somewhere to go. If you touch one end of a battery to your device, it's not going to work. You have to touch both sides. Why is that the case? Because in the battery, electrons come out one side, go through your device, and back into the battery. And if you don't touch both sides, the electrons have nowhere to go. And what electricity is flowing electrons. Like water has to have a place to go if you want to have a flow. So the electrons are flowing somewhere. In my analogy, they go to the positive side of the battery. In this case, the electrons have to go somewhere. Where do they go? Well, they hook up with some oxygen that you're breathing, these electrons that have been coming through the system, and hydrogens that are floating around in here. What that does is it makes water. It means the electrons found somewhere to go, and you can move more electrons through, pumping more hydrogens, and that is how we make ATP. So the electrons were used to move these hydrogens from here to up here. Let's go to the end part of this. Now we have a whole bunch of hydrogens. They want to get back down through the membrane, and the only way for them to come back through is through a molecule called ATP synthase. And you can see it's keyed right here in blue, ATP synthase. What does that mean? ATP synthesis enzyme. So as these hydrogens move, this entire enzyme rotates like a wheel. And what that rotation does is it causes the ADP, which is the discharge battery, plus a phosphate, become ATP. And notice now it has that lightning bolt symbol, which means we've made energy. So we took the energy from this big pool of hydrogen and converted it into a molecule that you can use to flex your muscles or do whatever you need to do. So the key points are NADH and FADH2 drop off their hydrogens and electrons, which are shown by the stars. There's the hydrogens. The moving electrons move through here, and as they move through each of these cytochromes, they pump hydrogens from the inside to the outside. The electrons have to have somewhere to go, so they hook up with some of the hydrogens, the oxygens that you breathe, and the electrons come all together to form water. Now, we have a whole big pool of hydrogens that have been pumped by those electrons as they flow back down through the ATP synthase that causes the discharge battery of ADP, plus a phosphate that's floating around to be reattached into this key molecule called ATP. So I may be asking you to tell me about this, and I'll describe how to do an how to do an essay in class tomorrow when I see you. So this is refreshing you on how we went to this. There's glycolysis, anaerobic, and then eventually we're going to make acetyl-CoA, which is the prep stage. Then we go into the Krebs cycle, making some ATP, or primarily making the hydrogen carriers, CO2 coming off as a waste. This is just a review of this overall. Okay, that is the lecture for today. It's weird not having any students here, but I'm glad you guys got a chance to hear it. Be sure to watch this as many times as you need to fill in your notes. Normally, I will be asking you guys to have your notes filled in before you come to lecture, and then that way I can move through it quickly and you don't have to slow me down or, ask questions. I mean, you can ask questions, but you don't need to slow me down because you're filling in the notes. Those should already be done. I hope to see many of you tomorrow in lab on campus, BCC campus. Until then, I'll say have a good night, and we'll see you again next week for lecture. Hopefully more people will show up. Have a good night, guys.